Hello to everyone, and welcome to today's webinar on the genetic counseling in patients with primary immunodeficiencies and immune dysregulatory diseases. My name is Ivona Aksentijevich, and I will serve as the moderator for this presentation. I'm a research scientist at the National Human Genome Research Institute in Bethesda, Maryland, and I work on identification of new disease-causing genes in patients with inherited systemic autoinflammatory diseases. These patients present with enhanced inflammatory responses and are typically seen by rheumatologists. In addition, I'm a board certified in clinical molecular genetics, and I run a CLIA-certified genetic testing lab for patients with these disorders. Today, we have two enthusiastic young ladies and genetic counselors, Mrs. Leandra Tolusso from Cincinnati and Mrs. Jennifer Holle from San Francisco, who will enlighten you on the genetic testing in patients with primary immunodeficiencies. Leandra is a licensed and board-certified genetic counselor at Cincinnati Children's Hospital, where she specializes in immunodeficiency, prenatal, and laboratory genetics. As part of her role, she partners with the providers in the bone marrow transplant and immunology division to provide genetic counseling services to patients with known or suspected primary immunodeficiency syndromes. She's also a clinical collaborator on the Cincinnati Children's Immunology Exome Slice reporting team. She loves the fast-paced and ever-changing landscape of immunodeficiency genetics, and is frequently amazed by how she learns something new in this field every day. Jennifer is a licensed and board-certified genetic counselor with 7 years of experience in clinical and laboratory settings. She currently works at the Invitae, a clinical genetic testing company, where she performs genetic variant interpretation, provides clinical consult assistance to ordering clinicians, and leads the immunology and hematology clinical team. In previous roles, she has provided genetic counseling to patients with primary immunodeficiencies and hereditary cancer. She loves working on the genetics of primary immunodeficiencies because of the incredible impact the genetic team test results can have for the treatment of patients with these conditions. Jennifer is a full-time employee and a shareholder in Invitae. Leandra has nothing to disclose. Leandra and Jennifer, please take it from here, and we would like to take any questions from participants at the end of their presentation.
All right, thank you everyone for being here today. So you probably don't need two genetic counselors to convince you that genetic testing is in the limelight right now. It's occupying a special place in science, medicine, in the news, and in your online shopping cart. We're exposed to genetics and genetic testing on a daily basis, and so are our patients. The use of genetic testing for diagnosing patients with primary immune deficiencies has been around for quite some time, but its widespread use has really exploded recently. Just this year, the CIS conducted a study of its members' use of genetic testing for diagnosing patients with primary immunodeficiencies. And among other things, the survey determined that genetic testing is appropriate and important for the majority of patients with known or suspected PIDs, and that genetic counseling should be provided to patients undergoing genetic testing, either by an immunologist or by a genetic counselor with experience in immunodeficiency. There aren't enough genetic counselors specializing in primary immunodeficiencies, and most immunologists don't have enough time to spend a full-length genetic counseling discussion with each patient undergoing genetic testing. But patients do have questions about genetics, and it's important that they understand how their genetic information relates to them. So we need to take a team approach when it comes to meeting the genetics needs of these patients. So throughout this talk, we'll share some insights from the front lines of genetic testing and genetic counseling to provide tips that will help you guide your patients navigate the complex world of genetic testing for primary immune deficiencies. There's a lot to cover about genetics, and we won't be able to do it all, but there are a few objectives that we'd like to focus on. First, we'd like to learn to gather an accurate family medical history that's really targeted to primary immunodeficiencies. We want to compare genetic testing types and understand their relative utility in diagnosing these patients, anticipate patient questions and feel prepared to answer them, and become comfortable with explaining genetic test results to patients.
All right, for this first section of the talk, we'd like to focus on the importance of family history and some helpful tips for making sure you're getting the best information. We all know that the patient's medical history is a very important part of their evaluation, and so it's important to include the family history in this portion. A family history of primary immune disease is often included as one of the warning signs for primary immune deficiency. Since some PIDs can affect multiple generations, it's important to take a detailed but targeted family history to help you determine the risk of a PID in your patients. However, it's important to remember that not all PIDs appear to run in families, and so a negative family history is still very informative. There have been a number of studies looking at predictors of PID, including a 2011 study that I cited here, and family history is often found to be one of the strongest identifiers of PID in children who are referred to immunologists. Taking a family history can come in many forms, depending on your specific clinic's preference. And for those who aren't familiar, this is a picture of a pedigree, which is the preferred method for most genetic counselors and geneticists. So in this picture, squares represent males and circles represent females, and each row of the pedigree represents a different generation. Individuals who are shaded in orange are affected with a certain disorder, and those with dots are carriers. This is a pedigree that shows the kind of family tree that we see in textbooks and lectures about PIDs all the time. It's abundantly clear that there is something running in the family and that it only affects boys, and it seems to be running through the female line in the family. This is a classic example of an X-linked recessive primary immune deficiency like X-linked SCID, and it's the type of family history that we would hope would be pretty striking in your clinics. But what if it's not so straightforward? This is the kind of family history that I see all the time in clinic. How do you begin to interpret this information? Our patient is denoted with the arrow in the yellow shading, and he has recurrent pneumonia and lymphadenopathy. He has a sister who has recurrent ear infections, a paternal cousin who is, quote, sick all the time, and a maternal uncle who had lymphoma. These relatives all have fairly nonspecific symptoms and things like ear infections, which are common in children, being sick all the time, which doesn't really give us any specific details to fully understand the severity, and lymphoma, which is a fairly common cancer. So how do we begin to interpret this? Is this a sign of an inherited PID in the family? Is this something in their environment? What is the magic trick to understanding a family history like this? Well, there isn't one, but without more information, this family history might not be the most informative, but still important to have because this could be helpful information later, especially once we've done more of an evaluation on this patient. It's important to keep in mind that a true understanding of the family history does go beyond just the genetics. Yes, genes are passed on from family member to family member, from generation to generation. So primary immune deficiencies can run in families, but there are other things that run in families, like shared behaviors and environmental factors. For example, nutrition, hygiene habits, exposure to secondhand smoke, and presence of animals in the home are all behavioral and environmental factors that are often shared in families and can impact how often a child gets sick. When faced with a family history like this, you might think none of these siblings or relatives are affected, so it doesn't look like something's running in the family, meaning it's probably not genetic. And that might be true, but considering that all the siblings live in the same home and go to the same school, they likely have shared behaviors and environments. So why is it that one of them is sick, but often and the others aren't? Why does the younger one's illnesses not seem to spread through the family like we so often hear? Once you eliminate these behavioral and environmental factors, it actually becomes more likely that this child has a genetic predisposition that's not shared by his siblings, either because a new mutation or something that his siblings didn't inherit. So this just shows that a negative family history is important too, and you can't just look at a child who does not have a family history and assume that it's not a genetic condition. Overall, the truth of the matter is that sometimes the family history can provide a piece of the puzzle that brings everything into clear focus, suggests a specific diagnosis, and can substantially guide testing and treatment strategies. But more often than not, it may be only one piece of the puzzle that, when all combined, makes the picture a little bit clearer. Either way, it's an important part of the diagnostic workup for patients with suspected primary immune deficiencies. So in order to get the most effective family history, you might be wondering what information specifically should you be looking for. So this slide is intended to give you some questions to think about when taking a family history. Asking, does anyone else in the family history have an immune deficiency is a good question, but it doesn't always get the complete information that you're looking for. Families might not be familiar with symptoms of an immune deficiency, so they might not know how to answer that question. Asking if there's other children in the family who have been sick a lot, or if there are people who've been hospitalized for an infection, are good ways of determining the likelihood that relatives have an inherited immune deficiency. And then there's other questions that aren't specifically about immune deficiencies, but can give important information. Are there any babies who died early on in life, especially if it was unexplained? Are there children who were born with birth defects or anybody with disabilities? Are there people with cancer in the family, especially things like leukemia and lymphoma? Are there anyone with autoimmune disorders? And these are all health questions, but it's also important to move beyond just health history questions and ask more about the family's background, especially whether the parents are related, so if there's consanguinity in the family, or what their ethnic and ancestral backgrounds are. These can be very insightful. It's important to remember also that all team members can gather and document this information in the patient's chart, and you don't just need a genetic counselor to be taking a family history. So to wrap up this section on family history, I wanted to give an example of a family in which the questions that you ask can help you go from a relatively unknown diagnosis to a very specific one. So here's a family history that you might take in clinic when you're evaluating a patient with bone marrow failure. Our patient, again with the arrow, is a newborn patient and has 2 healthy siblings. You ask the parents if there's other family members with bone marrow failure, and you find out that he has an affected second cousin. Based on this, the differential diagnosis is pretty wide. There are so many genes that can cause bone marrow failure, so where do you even start? And the question really is, does this cousin have the same thing, or is it just an unlucky coincidence in the family? The next question you can ask is there any consanguinity in the family? Do they share any blood relatives? And you find out that yes, the parents are related. In this case, they're second cousins. And you can now see that this cousin of our patient who has bone marrow failure is related through both sides of the family due to those shared blood relatives. So it does make it more likely that their diagnoses are related. And since we know that consanguinity raises the risk of an autosomal recessive disorder, that fits well and is consistent with many of the genetic causes of bone marrow failure. Next, you ask about ethnicity, and you find out that the parents identify as Ashkenazi Jewish. This information may be helpful, as you can then investigate whether there are certain bone marrow failure syndromes that are more common in this population. And finally, you ask about other major health issues in the family, and you realize that there's a strong family history of breast and ovarian cancer. So now you can put all this information together. You are looking for a disorder that causes bone marrow failure. It's probably autosomal recessive. It's more common in the Ashkenazi Jewish population, and it seems to predispose to breast and ovarian cancer in carriers. Fanconi anemia due to BRCA1 or BRCA2 mutations would fit all of these criteria. So now, after asking just a few more family health history questions, instead of ordering a large and expensive bone marrow failure panel, you may choose to order a targeted test of BRCA1 and BRCA2. You may also have identified a diagnosis in the family that could substantially impact other family members, and this was all just by asking a few more simple questions.
All right, so now after you've taken the family history and you have the personal history information, you might be ready to start thinking about what genetic test you want to order for your patient. So the purpose of this section is not to become an expert in genetic test technology, but rather to realize that there are benefits and limitations to most of the different types of genetic tests that are out there. And we'll take a look at some of the most common ones that are used to diagnose patients with primary immune deficiencies, and then look at a few example cases. But first, because it seems to be impossible for a genetic counselor to do any kind of presentation without including this slide, this is just a little context to make sure everyone's on the same page, that all of our cells contain chromosomes, which are like packages for DNA, and genes are segments of DNA, and they are made up of DNA letters, bases called nucleotides. So we're going to look at some tests that really focus on this DNA sequencing level, looking at those nucleotides, and we'll look at some tests that are focusing on the more broad genomic level of looking at the chromosome level content. Okay, so single gene testing is a good place to start the conversation because this is the original old school method of genetic testing. So single gene testing is, no matter how you do it, whether using Sanger sequencing, which is the old-fashioned technology, or the more common and newer next-generation sequencing technology, single gene testing is testing of a single gene. So this is a good test because it's fast, it's affordable, it limits unexpected findings. So if you're really just testing one gene, you're not going to find mutations in another gene that you weren't anticipating. However, it can also have a limitation because by choosing a single gene, you're making an assumption about the patient's phenotype and genotype. If you have made the wrong guess, if that's not the right gene and you don't find the mutation you're looking for, this can sometimes result in what we call serial genetic testing, where you test one gene and then the next and then the next. And this kind of cascade of genetic testing can be time-consuming and expensive and frustrating. Another potential limitation for single gene testing, which is really a little bit more specific to the Sanger sequencing methodology, is that it may not detect deletion and duplication mutations. These can be important, and we'll talk about these mutations a little bit more later. So the best uses for single gene testing are when a patient's phenotype is highly specific. So for example, if you have a male patient with agammaglobulinemia and no B cells, you should be thinking about the BTK gene. And this is a very good time to use a single gene test because this is a pretty specific phenotype. Sometimes you have flow cytometry results, which suggest a specific gene. So if you have tested the perforin protein and there's no perforin by flow cytometry, you might want to go ahead and just jump to the PRF1 gene sequencing. If you already know the mutation in the family, typically single gene sequencing is used for testing other relatives. It's used for carrier testing, maybe when you know that one partner is a carrier and you want to test the other partner for mutations in the same gene, or certainly when you're testing related bone marrow transplant donors. So the next test we'll look at is a multi-gene panel. So these are done by next generation sequencing technology. And these include multiple genes which cause a disorder. So they might be phenotype specific, smaller panels that are targeted towards something like 20 genes that are causative of HLH or ALPS or something like that, or they might be expanded. And the expanded tests these days can really include a lot of genes. So there are a lot of panels that include 200, 300 genes associated with primary immunodeficiency in general. So there's a lot of variety in terms of size for these panels. These tests are fast. They're getting more affordable. At this point, I would say they are relatively affordable. They can include many, many genes. They do detect deletions and duplications. However, you'll note that I listed this concept of deletion and duplications as both a benefit and a limitation to this test. And that's because some panels on the market do detect these mutations and some don't. Next-gen sequencing can detect dels and dupes, however, not all panels do. And I think it's really important that when you use a panel for patients with PIDs that you have del-dupe analysis included. Internally at Invitae, we've seen that about 8% of pathogenic mutations in patients with primary immune deficiencies actually are one of these deletion or duplication mutations. And that's a pretty significant chunk. So I'm always harping on this with people to order a panel that includes that analysis because it is pretty significant for this patient population. Multi-gene panels also provide excellent coverage. So what I mean by that is that if there are regions of the genes that are not sequenced very well, the lab will typically go back and fill that in to ensure that there's a certain level of coverage that's demonstrated. We also are able to target specific tricky mutations with this kind of panel. So if there's a deep intronic mutation or promoter mutations that you normally wouldn't be targeting, the lab can build in special ways to test for those mutations if we know that they're important. This is in contrast to something like the exome, which we'll talk about next, where you don't really have that same sort of depth of coverage. Multi-gene panels also support this concept that we call phenotype expansion. So this is something we talk about a lot in genetics right now, and you've probably been seeing this in the literature with primary immune deficiencies too. This is the idea that a gene that is known to be associated with a certain condition is later found to be associated with another condition as well. And when you order a panel that includes 200 or 300 genes on it, you can sometimes find these sort of unusual relationships where a patient who's presenting like condition A actually has a mutation in gene B that you didn't know would be associated. So that can be a nice addition to this test benefit. Some of the limitations of multi-gene panels, again, checking with your lab on whether or not it includes deletion and duplication analysis. Newly identified genes that are recently identified in the literature, they're hard to fold into one of these multi-gene panels at a frequent pace. So I would say that most labs are not able to update their next-gen panels more than maybe once a year or so. In contrast to an exome, where anything that's recently identified in the literature would be included in your analysis, this is somewhere where a multi-gene panel can lag a little bit behind the literature. And also similarly, if the mutation hasn't been reported before, or if the gene hasn't been reported before, it's not going to be included on that panel. So if there's a novel, if your patient actually is the first patient with a new gene disease relationship, we wouldn't find that on a targeted panel. And then the last thing here for a limitation, this is really something kind of general to genetic testing, and I think it's a really important concept, but the more genes you test or the more genetic material you test, the more uncertainty you open yourself up to. So I think that's something that's really important for patients and clinicians to know, is that when you order something like a 200 or 300 gene panel, you're going to get some variants of uncertain significance. And I would say that on our 200 gene panel, that is the most common finding we see, is we see some variants of uncertain significance in most patients. Okay, so these panels are best, the small multi-gene panels that are targeted are best for conditions that are multigenic, so like ALPS, where we know there are multiple genes that cause that condition. Large panels are best for patients with an atypical or unclear phenotype. They're also really good for patients who are very sick and need genetic testing quickly and can't wait for that kind of stepwise serial genetic testing. And I think it's important that patients should be prepared for some sort of uncertainty in their results. So that's something I think should be worked into the discussion about what to expect with this test. Okay, so now we're scaling up to look at tests that really work at the genomic level. So a SNP microarray is a test which assesses copy number losses and gains, or basically large deletions and large duplications, across the genome. So in the last section, I talked about deletions and duplications and how they should be included on your multi-gene panel. When I'm talking about those, I'm talking about small deletions that are under the size of a gene. So they're within the gene, they're a couple of exons, that sort of a thing. Here, when we're talking about deletions and duplications, we're talking about big multi-gene changes. So things where there are maybe 5, 20, et cetera, genes that are missing or duplicated in a patient. That's why this test is really great for patients with multiple congenital anomalies, where you expect that there are multiple genes that are causing different phenotypes that are missing or added in the patient. The other thing that's kind of interesting about this test is that it can detect what we call regions of homozygosity, or places in the patient's genome that are identical in the maternal and the paternal copies. This feature can be valuable when narrowing down genes of interest in families where you know that the parents are related by blood. So because this test is really large scale, it doesn't detect small scale deletions and duplications like you would on a sequencing panel. It doesn't detect sequencing mutations. It's not really designed to do that. That's just not in its technology. And because it is a large genomic level test, there are findings of uncertain significance. So sometimes we need to go back and then test mom and dad to see if they also have the same microdeletion, and if it's something that's common in healthy people or not. Okay, so microarray is good when you suspect a microdeletion syndrome. So like DeGeorge or 22q deletion syndrome. If you have a patient with developmental delay, autism, multiple congenital anomalies, or other syndromic features, this is a great test because you're expecting there to be multiple genes behind those kinds of findings. Or if you suspect consanguinity, or if you know there's consanguinity in the family. Now, it's not important to test every patient who has consanguinity in the family with a microarray. This can be valuable if you really have no idea where to start with the genetic testing and you want to narrow it down to those regions of homozygosity. But if you know the patient has, X-linked agammaglobulinemia, you don't need to do a microarray to diagnose that. That's really when you are kind of grasping at straws and looking for somewhere to start. So for Microarray, the family should be aware during the process of consenting of the possibility to detect consanguinity. That's important. Some people know about that in their family, and some people don't, but it should be discussed beforehand. And there's also a possibility of follow-up parental testing, because sometimes that's the next logical place to start when you have a variant of uncertain significance. Okay, so next, everybody's favorite test, whole exome sequencing. So this slide is just for context to compare and contrast a genome and an exome. So as you all know, the genome is all of the DNA, all of the genetic material in the body. The exome is focused just on the coding regions of the genome. So this is about 1% of all of the genome in total. Okay, so the benefits of exome sequencing are that because you're testing typically mom and dad with the patient sample, you can detect what we call de novo or brand new variants that are only present in the patient. We can also, because we're testing mom and dad, detect the phase, so you can tell if two mutations in the same gene are on the same or opposite chromosomes, and that can be useful for the analysis. You can detect multiple genetic causes with an exome. So if a patient actually has two distinct genetic disorders, you can find that with this kind of a test. Like I mentioned before with next-gen sequencing panels, exome sequencing is current with the literature. So if there's a brand new paper that's describing a new genetic association, the exome sequencing analysis process should pick that up. So this is a concept that I threw in here a lot and tried to highlight it so it's really clear. Exomes can support gene discovery, which is the idea of finding that first patient who has a new gene, a new phenotype that's never been described before. However, this is specific to research exomes. A clinical exome, like one you would get through Invitae or some of the other genetic testing labs that are out there, aren't going to find a new gene disease association that's never been described before. It's not really what they're targeted at doing. Research exomes, however, can. Some of the limitations of exome, there's so much material that you're testing here, there's so much analysis, so it's relatively slower, it's relatively more costly, although the price of this testing is dropping. The cost is really complicated because so much of it depends on insurance, and Leandra is going to talk about that some more in her slides. Even though we're testing so much genomic material, the positive rate for this test is still kind of hovering around 20 to 30%. So that means the majority of patients aren't going to find an answer from this test. And I think that's really important in terms of setting appropriate expectations for exome sequencing. Exome sequencing has lower resolution than a panel. So before, when I was talking about how if there are tricky promoter mutations or mutations that you know are specific to that gene, an exome usually is not going to be baiting those or really targeting those regions on their test. It's broader in scope, but there's less depth to it. So there's less resolution overall. If you really suspect a certain condition for a patient, and if you know that those genes are out there, are known, it's better to do a panel because of that resolution issue. Sometimes I've even had people contact me and say, hey, I've had an exome, it was negative, but I really think this patient has SCID, and then they've gone back and done a SCID panel after the fact. There is a risk of incidental findings, or I'm sorry, I'm going to start with uncertain results. There is a real risk of uncertain results. Because there's so much material here, there's a lot of fusses, we call them variants of uncertain significance. I would say that this should be the expectation. This is the norm to get some sort of uncertain results out of this kind of a test. Now, incidental findings could be a whole webinar in and of itself. This is a huge topic. It's something that we discuss all the time in genetics, and this really complicates the consenting process for this test. But this is the concept of finding off-target things you weren't looking for. So for example, finding a risk of breast and ovarian cancer or heart disease in a 5-year-old patient, something that you really weren't intending. That opening yourself up to that kind of information takes a lot of thought and conversation on the part of the clinician in the consenting process. So that, I guess, is a limitation of this test, that there's kind of a lot of burden in terms of getting patients ready to undergo it. But exome sequencing is really great for patients when you suspect multiple genetic causes. So maybe their phenotype is so strange because they actually have 2 different conditions. This test is great for that. If you're doing a research exome, you actually can find a new novel genetic cause for patient symptoms. If you've ruled out all the other likely diagnoses, exome might be the next, kind of final step to go to. If you know that you think the patient has a certain condition, start with that. But if you've ruled everything else out, then the exome is really kind of the best we've got right now. And it's also best used when parent samples can also be sent. So this isn't a requirement. You can send just a proband, patient only, or you can send the patient and a parent. But if you can send mom and dad also, that's really, really great. So again, families should be aware of unexpected findings, and the clinical exome doesn't support gene discovery. The conditions there. Okay, so this is your screenshot alert. This is a nice resource. I'm not going to go through all of these pieces here, but this is a nice comparison if you want to go back to it and reference it later, of benefits and limitations of these tests. Okay, so now is your chance to practice. So we'll look at a couple of cases here. So our first case is a 12-day-old baby girl who's referred following an abnormal newborn screen for low trex, so suggesting a T-cell deficiency. She has short femurs that were identified on prenatal ultrasound, and her family history includes several uncles and cousins with what the family describes as dwarfism. They are not related by blood, however, the family is Amish on both sides. So in this case, what is the best test? So a single gene test is the best one for this example, because there's a gene called RMRP that really unites all of these features that we're looking at in this patient and her family history. So RMRP is associated with a recessive condition called cartilage hair hypoplasia syndrome, and this is associated with T-cell deficiency, short stature, among other things, light-colored, sparse hair, and there's a very common mutation in the old order Amish population. And that was the situation in this patient's family. She was homozygous for this mutation, and that was able to be detected with a single gene test. Okay, the second case. Here we have a 9-month-old baby boy who was born premature. He has developmental delay, anemia, chronic granulomatous disease, hepatitis, hearing loss, and nystagmus. So in this case, there's a lot of stuff going on. Which is the best test here? Here we would go with a microarray. And we would pick that because the patient has developmental delay, has multiple organ systems affected, but without an apparent unifying cause. And when we tested him, that's exactly what we found. He has a microdeletion syndrome affecting the X chromosome. That includes the X-linked gene for chronic granulomatous disease. It includes an X-linked retinitis pigmentosa gene, as well as several others that explain his developmental delay. So I think it's important to realize that it is tricky to sometimes choose the right test for a patient. And one of the things that I want you to know that genetic counselors can help you with is which test is the right one for a given personal and family history. You have colleagues in your institutions most likely who can help weigh in on this, and also you can always call your genetic testing laboratory. We're happy to help you walk through these kinds of considerations when you're thinking about ordering a test.
All right, so at this point, you have finished your initial evaluation for a patient, you've decided that genetic testing would be the best next step, and you spent a lot of time thinking about what type of test would be the best for your patient. Now you've made your decision. So it's time to talk about the patient and their family about that test. So I facilitate genetic testing for patients on a regular basis, and I've noticed that there's a number of common questions that seem to come up often in this population from the patients and more often their families and their parents, since we mostly deal with pediatric patients. So in this upcoming section, I'm going to talk about some of these common questions that you might hear and start talking about ways to answer them. So first off, most patients' questions will come up during the consent discussion for the genetic testing. As we all know, health literacy is pretty low in the general population, and I think it's even lower for genetic literacy. Most patients don't have a personal history or any personal experiences with genetic testing. At this point, many patients do come in with some history for things like ancestry testing, or at least they've heard about it in the media. But they may have misconceptions about genetic testing, especially on a clinical basis, and questions about how this specific test will work. So often when I ask what questions that a family might have, the response that I get most frequently is, I don't even really know what I should be asking. So it's important to have a conversation about the genetic testing with the family before you order the test to help manage expectations and limit confusion about possible results. And this conversation should include personalized information about the reason for testing. So for this family, what genes are we testing for and why do we want to test these genes? How will this test specifically impact the medical care? So overall, why is it important that we do this testing and why are we recommending it? Families also often want to know what kinds of results that they can expect from the genetic testing, so you should be prepared to discuss the meaning of the possible results. So most people are familiar with positive and negative results. So positive, meaning that we find a mutation or more than one mutation in a gene that explains the patient's symptoms, that diagnoses a specific genetic condition. And at that point, you can discuss what the health problems are that are associated with that, if there's other organ systems that are affected, what treatment options we might have, and those kinds of things. A negative result is the complete opposite of that. We did a test and we did not find anything to suggest that there's a specific genetic condition going on. As Jennifer discussed, depending on the extent or how broad your genetic testing is, you might stop there or you might be looking at more genetic tests in the future. Then the third result, as we've talked about already, is the variant of uncertain significance. And it's also my experience that the variant of uncertain significance should be the rule more than the exception when it comes to ordering broad testing for patients with primary immune deficiencies. When you're ordering a large panel or an exome, my experience is that the majority of patients are going to have some kind of variant of uncertain significance. And many patients aren't comfortable or aren't aware that we can be ordering something that doesn't have a black or white answer, that there's this gray zone in between. People think either they do or they don't have a genetic condition. And so it's important to explain that we might be facing some uncertainty. Sometimes genetic tests raise more questions than they do give answers. And that's just the expectation here. And I think it's a lot easier to manage that if you've had the discussion beforehand that this is something that's possible, rather than trying to backpedal afterwards and say, okay, we did this test and we found this thing that we don't really know what it means. And now we have to talk about that. So I think it's important to bring that up right out of the gate before you even order the testing so that patients have appropriate expectations of what to expect. If you have the information, giving the chance that they'll get a positive result can be helpful. So the expected yield of the testing. For some conditions that are well known, there's good numbers out there about what that is. But for some of these broader tests, we don't always have that information. But if you deal with a certain patient population on a regular basis, you can even talk about your experience and how often patients seem to be getting positive genetic tests. So Jennifer kind of talked about incidental findings, and families are often concerned about finding out information that might impact other family members or other health risks. So these incidental or secondary findings that are not necessarily related to the indication for testing. So a good example of this in the PID world is the ATM gene. So ATM is on a lot of the different large panels because it's associated with ataxia telangiectasia, which has immune deficiency symptoms. But carriers of a single ATM mutation can have an increased risk for breast cancer. And so depending on the test you're ordering, there might be a gene like this on the panel, and there might be a chance for an incidental finding. I'll admit that when ordering a 300, 400 gene panel, it's just not possible to go through every single gene, look it up online, Google it, and find out what incidental results are possible. So I give a general broad statement to families that whenever we do genetic testing, there's the chance of learning something unexpected. Our genes are in all of the cells in our body and they can have wide-ranging impacts. So it's possible that we'll find something that gives us information about other health risks that might not seem related but could be important for your family. If something comes up in the family health history, so like the one I presented earlier with lots of breast and ovarian cancer, you can directly address that and whether or not we might find something related to that. But for a family with a negative family health history, it's just good to give that broad statement that we might find something unexpected. We've also talked about the chance of getting information about the biological relationships in the family. So especially when doing testing like exomes, where you're taking mom and dad's DNA at the same time, we can find out things like non-paternity or that there's a blood relationship in the family that they didn't know about. So overall, there's no one-size-fit-all answer for how to do this pre-test counseling for each panel or each test, because they're all going to be different from each other. But it's important to review the test that you're ordering and make sure you know if there's any of these tricky little things on there so that you can bring that up with the family. Now, I'll say that the next question about insurance is probably the most common question that I get these days, both from patients and their families, but also the other providers that I work with. We have this genetic test, we want to order it. What's the patient going to have to pay? Is their insurance going to cover it? And again, there's no one-size-fits-all answer for this, and I could probably fill a whole hour talking about it, but the general answer is, I don't have a good general answer other than it's going to depend on your institution. So first thing to understand is, how does your institution bill for genetic testing? Do they request institutional billing, which is where the lab you send to bills your hospital and then the hospital bills the patient? Or do you directly send your tests to the lab and have the lab directly bill the patient or their insurance? The main things that this will affect is a lot of places that do institutional billing have a upcharge for the genetic test. So the list price on a website might not apply because your institution might add additional charge onto that. And then also it might affect whether it's in or out of network for your patient. So depending on their insurance and the lab you're sending to, the lab might be out of network, but if you do institutional billing, you might be in network. These are things that are complicated. And overall, I'll say that if you do institutional billing, I'd recommend talking to your billing representatives at your hospital or talking to your authorizations department and finding out, what information they can give you. If you send directly to the lab to do the patient billing themselves, they can often handle a lot of this for you, but they have great people who can answer your questions. So not only does it matter how you send the testing, but your patient-specific insurance obviously is going to make a big difference. Not all insurance companies cover genetic testing in the same way. So one company who accepts a test and will cover it doesn't mean that all will. The more you do this, the more you realize that there's certain insurance companies who are more notorious for being difficult and certain who let things go through a little bit easier with regards to genetic testing. So that just comes with experience. But some insurance providers have specific requirements. So there are certain companies who require that you pre-authorize the test, or else they'll just outright deny all of the testing requests. Some require that the patient meet with a geneticist or a genetic counselor before they'll cover any kind of genetic testing. And then some have other requirements for documentation. There's a few that require an actual 3-generation pedigree be in the chart. So you should be prepared to understand these things. You can't know all of your patients' requests, all of your patients' insurance companies' requests. There's just too many insurance providers out there. But as a general rule, for a patient who has a commercial insurance, I always recommend pre-authorization. Sometimes we get back a quick answer that says, no, you don't need to do that. This is a covered code, and that's great. But I'd rather be careful up front than just risk it, put it through, and then have the patient receive a large bill at the end. So when talking about insurance, you should be prepared to provide CPT codes, ICD-10 codes, and letters of medical necessity. Depending on your clinic flow, the families might be doing the pre-authorization, or you might be assisting with that. That's going to be different for everybody. But having this information, even just to provide it to the family, can be helpful. And then, as I've kind of mentioned, if you're ordering straight through a genetic testing company without going through your institution specifically, then some of the labs will have billing folks who can help you out with this. And since I mentioned letters of medical necessity, and I write a lot of letters of medical necessity, I figured I'd provide some tips. This is another warning. You can take a screenshot of this. I'm not going to go through all of these things here, but this is some specific language that I use in my letters of medical necessity that I find to be beneficial. In general, if you're requesting pre-authorization or you're writing an appeal or whatever it is that you're doing, that you're providing a letter of medical necessity, you want to be as specific as possible about the genetic test. So writing, this test will change medical management, is not specific enough. You should provide as specific examples as you can. So if you're considering bone marrow transplant based on the results, put that in your letter. You should also include a list of genes of interest and why this test is the best test. So often when I'm ordering large panels, I'll put my list of genes of interest. So say it's an HLH panel, I'll put all of the HLH genes on there. And then I'll say in my letter that if we don't get this panel, I'll have to order all of these by single gene testing, and this is the price for single gene testing at the lab that I'm ordering from. And this is why this panel is better for this patient than ordering all of these tests individually. And then if you have scientific literature that supports your testing strategy, it's always good to provide that information to the insurance companies. Just a quick PubMed search can usually help you with that. There's becoming more scientific literature on the broad tests like Exome and large panels, so that's becoming more helpful. But certainly if you're ordering a single gene or a smaller test, there's some pretty good literature out there that will help support it. And then as a general rule, whenever I write something that looks really good or that an insurance company really likes, I always save it and I use it again and again, changing in the patient's information, specific information, because there's no reason to reinvent the wheel every time you want to write a new letter. So going back to patient questions, a really common one that I've been getting more and more recently has been about privacy and how their genetic data will be kept private. My sense is that there's been a lot of news coverage recently about genetic testing data, especially from direct-to-consumer companies being shared with third parties or with law enforcement agencies. And you've probably all heard stories about people being caught for crimes that, because their cousin had genetic testing through a DTC company. And so patients are increasingly more aware of their privacy and are asking this question. So it's good in general for everybody to be familiar with the Genetic Information Non-Discrimination Act, or GINA. This is a law that protects against discrimination based on genetic testing results for health insurance and most employment. So that means health insurance companies can't make coverage decisions or premium decisions based on genetic testing results. They shouldn't even really be asking for it. And that employers can't hire you, fire you, change your salary, change your benefits, or anything like that based on your genetic status. Notably, though, this GINA law does not include protections for life insurance, disability or long-term insurance, and military work. So I always tell patients that, if you're considering a career in the military, it's important to know that this is something that they can ask for. And especially when it comes to pre-symptomatic testing in other areas of genetics, life insurance can be a big consideration. It's also important to be able to compare the direct-to-consumer tests with clinical tests. So you should be ordering your testing from a clinical laboratory, and these laboratories have to abide by the HIPAA requirements. So the privacy of the data is probably safer with these laboratories than with direct-to-consumer companies. The direct-to-consumer companies don't fall into HIPAA requirements. So they'll have a privacy policy. Usually it's like a terms and conditions that you have to opt into on their website that, if you read through all 40 pages of it, you'll understand their privacy requirements. But because they don't have to abide by HIPAA, it can vary from company to company, which is why we're seeing that media coverage of people having their information shared. We did want to mention that clinical laboratories may be sharing some of the information, but it would be de-identified. So it is common for many of the laboratories to put de-identified information into public genomics resources. A common one is called ClinVar. And this is where they can share specific mutations and what the call was on those. So was it a pathogenic mutation, likely benign, a variant of uncertain significance? But none of this is going in with any identifying information on the patient. And then some labs share to rare disease programs that aim to connect providers and healthcare providers who are caring for children with the same rare diseases to hopefully better understand these conditions. And finally, an important question lots of people ask me is, are they doing research on my genetics? Are they using my DNA for research? And the answer is probably for that. It depends on the company, and they should all have information on their consent forms or on their test requisitions about their research policies. The important thing here is that some of the consent forms have an opt-in checkmark, so check this if you want to be part of research, whereas some other companies have opt-out, so check this if you don't want us to do research using your sample. And so it's just important to know if there's a lab that you order from more frequently, just read through their forms so that if your patients ask you about research, that you're well prepared to answer kind of the specifications and what the testing company is doing from that perspective.
Okay, so in the next section, we're going to switch gears a little bit and look at the genetic test results and how to interpret them. So Leandra did a nice job of kind of talking about how you would consent a patient for the types of expectations of results that you would have. And a positive result, of course, is what everybody is hoping for, the answer to the mystery. This is pretty straightforward. So in this example, we have a positive result where there are 2 pathogenic variants identified in the same autosomal recessive gene. In this case, this finding explained the patient's phenotype, so we'd say it confirmed the diagnosis. You could proceed by discussing the risk for other siblings to be affected with the same condition, as well as reproductive risks for this patient's children, the parents, and other relatives. A negative result is a little bit less straightforward than it at first seems. So this test did not find the cause for the patient's symptoms. However, it doesn't rule out a genetic etiology for them. So there could be a mutation that was tricky to detect with the technology that we have, like maybe it's an inversion mutation or something kind of wonky that normally you wouldn't pick up. So that could be something that could explain a negative result. There also could be a gene outside of the panel of genes that you tested that explains the patient's phenotype. So a negative result doesn't necessarily mean your odyssey is over. A variant of uncertain significance. Of course, everybody's seen this, everybody hates these. But basically with the VUS, we don't know if this finding is a benign variant or a pathogenic mutation. There are some clues that we can look into to try to figure out how much we care about this finding. So we would look at the inheritance pattern, we'd look at the gene phenotype association. So if this variant in an X-linked gene is found in a male, it's more significant than if it's found in a female. And if the phenotype matches, that's more significant, of course, than if it doesn't. So now we're going to look at what I'm calling a complex result. So this is pretty typical, where you find one variant that's pathogenic or likely pathogenic in a recessive gene. Unfortunately, this recessive gene takes 2 pathogenic mutations to cause the phenotype, so this doesn't really explain it. This isn't enough of the answer, this one finding. And then there are also 3 variants of uncertain significance here. So from the front, they all look equal. You know, there's a variant in STAT1, MOGS, TTC7A. But we can use some of those clues about the inheritance pattern, the associated phenotypes, and think about whether there's any follow-up testing that might be needed to help tease apart which of those VUSs are more important than the others. Oops. Okay, so delving deeper into that example, in this case, we have a patient's presentation. So this was a 5-year-old patient who had persistent elevated inflammatory markers, recurrent infections, autoimmunity, and chronic mucocutaneous candidiasis. There's no family history in this patient. So looking at that TMC6 variant, that's the likely pathogenic variant, but there was only one of them, didn't explain the phenotype here. That's not associated with what the patient has, so we can pretty much rule that out. This means that the patient's a carrier for that condition, but that's not most likely causing the patient's symptoms here. There are 3 VUSs, and by looking into what these genes are associated with, we can see that STAT1 is associated with chronic mucocutaneous candidiasis. The other 2 aren't really relevant to the patient's presentation. STAT1 is also the only one here that's autosomal dominant, so one variant, if it's pathogenic, actually would cause the phenotype. With the other 2, we would need another variant as well. So now I want to look even a little bit further into these VUSs. And the reason I want to do this is to show you kind of what some of the information on the inside of the laboratory about this VUSs looks like. So first we'll look at the MOGS one, at the MOGS variant. So this variant is rare in control populations. It hasn't been reported in the literature. And the in silico protein predictors, which I will say are really not that valuable when it comes to variant interpretation. And I would never classify a variant based on these alone. They are conflicting in this patient. So we really know nothing about this variant. It's just a garden variety VUS where we don't know anything. The TTC7A variant is actually inclining towards benign. So it's rare in control populations, hasn't been reported in the literature. The protein predictors are benign, and this missense change has been seen in multiple mammals. So you might think that if this variant is, if it's common in different mammalian species, that it probably isn't as likely to cause disease in a human. Those aren't perfect, those are kind of secondary evidence, but it does sort of inkle towards benign here. The STAT1 variant has a very different story, so it's absent from control populations. It has been observed in patients, but the papers that we saw it in were really nonspecific, so maybe they reported this variant in a patient with primary immune deficiency, but not specifically in a patient with chronic mucocutaneous candidiasis, for example. There were some functional studies about this variant, which actually supported it being probably pathogenic, but we can't classify a variant based on functional studies by itself. We do also know that a different amino acid change at the same position is pathogenic. So that is pretty suspicious altogether. And this is something where if you saw this on a test report, this information is always written out on the report. And you can also always call the laboratory. If you have a variant where you think based on the phenotype, that's really suspicious, I want to know more about that variant, you can call and ask us this and we can help you work through that. In this case, that's what the clinician did. They said, hey, this STAT1 seems like a dead ringer for my patient. Can we test parents? And we did, and neither parent had this variant. So now we know that the variant is de novo in this patient, and we are actually able to reclassify it as pathogenic. Okay, so briefly, I want to look at what we're calling weird results. So these are things where you might see the words mosaic, possibly things that are a little bit unusual on your test report. So there are a number of phenomena in primary immunodeficiencies which can impact the expected allele balance that we see when we're doing the test. So the allele balance is like if the variant is heterozygous, if it's homozygous. Sometimes you can see that the variant kind of looks like it's less than heterozygous. And that might suggest that the variant is what we call mosaic. So the concept of mosaicism, and it's illustrated in this slide here, where a patient has a mutation that arises very early in development, but not so early that it was in the egg or the sperm. So some of the patient's cells, as they grow into a child or an adult, have the variant, but some cells don't have the variant. So some parts of the body might have it and some don't. This is pretty weird, but there are some autoinflammatory syndromes that are associated with this frequently. There's another concept called somatic reversion, which is even weirder. It's almost the opposite of that, where there's a variant in all the cells of the body that then disappears in a certain cell line, like say in the patient's blood. Sometimes if you're testing a blood sample for a patient with that going on, you would actually find maybe one mutation, but not the second one, or you find one mutation at a really low level allele balance. And there are also some conditions like ALPS where we know that there are mutations that impact only the double negative T cells. So if you're testing a blood sample, it might look mosaic because we're only getting that mutation in one cell line out of the blood, but it's not in all of them. So this seems really weird and crazy because these things are really weird and crazy. And if you get one of these types of results from your patient, don't panic. You can always call the laboratory and we can walk you through what might be going on here or what kinds of tests you should do to help follow up for your patient.
So I think for the sake of time, we'll skip through these sections. There's nothing particularly crazy about how to explain test results to the patient. Just make sure, you explain what is the result, how this might impact the patient's care, their specific treatment options that you're going to be discussing, and then impact for the family members is important. Are there other family members who are at risk for this condition, and how can we test them? So just want to wrap up here before we take some questions, hopefully this gave you some tools to help discussing genetic testing with your families and your patients. But if you need more help is available. So if you'd like to refer to a genetic counselor, if you don't have one at your institution, you can go to the NSGC or the National Society of Genetic Counselors website and use the Find a Genetic Counselor tool. There are people who do this in person, or there's even telemedicine options. And you can sort by specialty. For example, if you need a prenatal counselor to talk about reproductive options, you can look for something like that. If you have questions about specific tests, don't forget about your colleagues. They'll have very important insights. And then you can always call the laboratories. They have genetic counselors usually on staff who can help answer your questions. So at this point, Jenny and I will take some questions from everybody. And thanks for having us.
Thank you very much, Leandra and Jennifer, for this excellent, insightful, and detailed presentation. We have one question by a participant. And this is a tricky question. Will gene sequencing always pick up mosaicism if mosaicism is present?
That's a good question. I would say no, it won't always pick it up. And I think next-gen sequencing is more sensitive at detecting mosaicism than some of the other technologies like Sanger sequencing, for example. It really depends on the amount of mosaicism. So if it's very low level, like 5% mosaicism in the sample, that's probably going to be below the cutoff for the laboratory to report that finding. If it's present at like 25 or 30%, then you would report it. But this is actually a question I got the other day on the phone from a clinician who was looking for a mutation that was probably only in a certain cell line. And so in some situations, you might need to separate out the cell line of interest first and test that sample directly before just testing the blood in general.
So basically, you would pick up mosaic mutations by using either targeted gene panels or whole exome sequencing. That's the best way to find them. And even there is a question whether this mosaic mutation will be detected, depending on particular part of the gene, for example, is covered, by enough reads so that you can detect these mosaic mutations. Are there any other questions? We are running out of time, and I think we should wrap up this presentation. Yes, the next question is, how do you know it is mosaicism or just somatic mutation?
This is so hard. So when you see a possibly mosaic result, there could be a number of explanations for it. So the patient could be mosaic in all of their cells, including their germline. They could be mosaic in just a certain cell line, like just in their blood. And sometimes you can tease these things apart by testing other family members. So if you find the variant present in a parent, then you can say, okay, this is actually affecting the germline. Or if you find it, if the patient is an adult, if you find it in their children, you could say that it's affecting their germline as well. But you can't really know the extent at which mosaicism is affecting the patient's body without testing lots and lots of different samples, body tissue types. So I think there's a lot of uncertainty with this concept, and it makes the genetic counseling very difficult.
I would also just like to add that in these situations, one should really go after research labs. For example, we do this type of for patients without inflammatory diseases, where we'll have to get whole blood samples and then sort them out into different cells and do sequencing in particular cell types. So this is more really for research labs to help you answer this question. Are there any other questions? If not, I would like again to thank both of our presenters, Jennifer and Leandra, for the excellent job, and all who participated in today's webinar. Bye.